cold room. I'm running my first blue native page gel. So native page is a way to separate like protein complexes, keep them together and like separate them, as opposed to Sds Page where you're denaturing and unfolding all the proteins. The native refers that you're using them in their native state, so they're all folded up and they can be hooked up to other things and stuff. So you can tell if proteins are like forming complexes and stuff. And so I want to tell you more about it, but I want to do it outside of the cold room because it's cold in here. Imagine you have a protein complex, like say a trimer where you have 3 proteins stuck together. If you were to run this on Sds Page, what happens is you denature the proteins first. So you unfold them into like linear chains. So when you do this, you're separating the proteins. The reason why you do this is so that you can then separate them by size. So basically you send them traveling through a gel mesh. And so the proteins are going to kind of like slither through this mesh. This worked better in theory. So they're going to kind of slither through here. And the bigger, longer proteins are going to get tangled up more. So you're going to be, these little guys are going to travel easily through. Whee! They're going to go like, they're not going to get tangled as much, so they can travel faster. And so then when you turn off the energy, when you turn off the electricity, so basically you get them to move through the gel by applying electricity. So you have an electrical gradient where you have a positive charge at the bottom and a negative charge at the top. And the SDS, so the detergent that we're using to help denature, so help unfold these proteins and keep them soluble and happy, it's negatively charged. So it gives all these proteins a negative charge so that they'll get, they'll have an impulse to travel through the gel. They'll be tempted to travel through the gel. And then they'll be impeded by their length because the long ones, remember, are going to get tangled up. And so then when you stop it and you stain it, you're going to see bands corresponding to the locations based on their linear length. But you don't get any, so you get information about the relative sizes of the proteins that were in a mixture that you put in. But you don't know if the proteins that you put in were like part of a complex or whether these were just like separate free-floating proteins to hang out with. To get that sort of information, you can do native page. So native page, the native is for your separating complexes or proteins like in their native state as opposed to denatured. So we're not unfolding the proteins, we're going to run this together. And so when we run this together through a gel, now it's going to travel as a gel and it's going to, it's going to travel as a complex. And so its speed is going to depend on the size of this complex. And because you can have like these complexes with various different sizes and you're not like slithering, you're kind of all like, now it's depending on your shape too, instead of just like your length. Now we talk about different, like proteins have different shapes, and so the shape can also influence like how they'll move. Like are they really, really compact in a little tight little ball that can just whoo, or are they big and floppy and they're going to travel really slowly? But typically what we do is we have like a gradient gel. So like the mesh would be tighter at the bottom and looser at the top. And this way the complex will travel until it reaches the size where like the pores, so the holes in the mesh gel, the gel mesh become like too constrictive, and so it can't move further. So then you have your complex. So there are some things that you can do different things. So you can do here, like if they were complex, so if they were joined together, proteins that are joined together will travel together. Proteins that were unrelated, like not attached or anything, will travel separately. So you can then do several things. So one is you could just stain the gel like you would for like a normal Sds page just to see the location of the proteins. And so you would expect that if you had like a binding, if you were to run the proteins alone and then the proteins together, you would see an increase in the like relative size of the complex. If you have them complex together, you'll see like a higher up band. Compared to when you run the sample, when you were to run the proteins alone. But remember that the shape can also influence things, so it can get kind of tricky. You can do things like amino blotting, so use like a Western blot to use specific antibodies if you have like a suspicion about what various proteins are there and you want to test and see if they're there and like if they're in the same whatever. But be careful when you're using antibodies. They might, if you typically use like, so sometimes antibodies are these little proteins that recognize like specific parts of specific proteins, and so you can use them to see. So if you have like a labeled antibody, you can then see if you can like probe your gel and see if a specific protein is there. Those antibodies are often developed for use with like Sds Page. So in that case, they might recognize like parts of the protein like in the that are present in the linear form, like that are accessible in the linear form but not in the folded up form. So they might not work if you do it with like the first, if you were to probe your first dimension. You can like isolate these bands and stuff if you want, and then send them for like mass spectrometry. You can like elute them out. There's a bunch of different things you can do to try to figure out what the exact proteins were in a mixture. So what if you wanted to actually like, so sometimes if you like start with purified proteins and stuff, you can like do that. But what if you just want to see what complexes are like naturally in a cell or that sort of thing? So here you can combine, you're gonna have a lot more proteins when you're starting, and so you're gonna wanna probably separate them better. So what you can do is you can run the native page. So you run a, like separate the complexes by their size, and then what you could do is you could actually like cut out the gel bands, like so you load them in a well, so they're going to be like in a column. You can cut that out, and now you denature it. So you like soak the gel in SDS, so the detergent that's going to help denature them, give them a negative charge, keep them soluble and happy. So you can now take this denatured gel, turn it, and you kind of run like a normal SDS page gel below it. And now your proteins that were in the complex, they're going to run in the same column, whereas proteins that were like outside of the complex, they're going to be like in a different spot. But you don't have lanes here, so you kind of end up with like this weird looking like dots everywhere type of thing. Proteins are basically long chains of amino acid letters, and they fold up, these chains fold up into these pretty shapes because of the properties of these letters. And this also helps their shape and the various options that these letters provide also allow these proteins to sometimes form complexes with other proteins. And so we often refer to this as like quaternary structure, where you have multiple protein chains linked together or like working together. And so sometimes these chains can be, so sometimes a protein's functional form will be a multimer. And so a multimer is when you have like multiple protein chains together. And so sometimes this will be like the same chain. So it would be like a like a homodimer or homotrimer or homotetramer or whatever, or they could be different. So that'd be like a hetero. But so there are different types of this. So sometimes they're like tightly bound together, and it's really this functional unit that needs all of them. And other times you can have more transient interactions between proteins. But in either case, you might want a way to be able to tell if these proteins are forming like stable complexes. And so one way that you could do this, or to identify like what a protein is complex with. One way that you can do this is with native page. And so with SDS page, what you do is you take the proteins and you're going to denature them and run them through a gel, and they're going to separate based on their length. With native page, you're going to keep these complexes together. And so the complexes will run together, and so they'll run like a bigger thing. And so they'll show up as a higher band. So this is the basic idea of where we're going, but let's look at it a little more in detail. So with SDS page, we're using a denaturing agent. So we're using heat to help unfold the proteins, and then we're using SDS, which is this detergent. And so detergent's just like an artificial soap. It has this long hydrophobic or lipidy part, this is going to glob onto the hydrophobic or water excluded regions of proteins. So when proteins fold up, they tend to like hide their hydrophobic regions. So the regions that water excludes, they hide those in like the center of the protein away from the water. And so when the protein unfolds, you're exposing all those hydrophobic regions that the water does not like. And so this can cause the proteins to kind of like aggregate together. So instead of like folding up nicely, they'll just like clump together and like precipitate. And so like just like come out of solution. So SDS is going to help with this. It's going to, this like hydrophobic, this long nonpolar part is going to glob onto those hydrophobic regions of the protein and prevent them from like globbing together. And because it has this negative charge, it's going to help keep the protein, keep it linear, and give it a negative charge. And so you can think about these negative charges as kind of helping prevent these from clumping too, now that you just have a ton of hydrophobic stuff, right? That's not what you have. Instead, because you have those negative heads, you're going to get this like linear protein product with a negative charge. And the charge is going to directly relate to the length as well as too. So you kind of like have this constant relationship between the charge and the length, which is important, as we'll talk about later, because proteins can have different charges. And so this kind of wipes the slate clean. And now you're just separating proteins based on their length. And how you do that is using electrophoresis. So Page stands for polyacrylamide gel electrophoresis. And so polyacrylamide is this chemical that makes this gel. And so this gel, it's like this meshy thing. And the longer things are going to move through the mesh, and they're going to get like tangled up. And so the bigger things are going to move more slowly. And so you can't see them move when they're moving through the gel. You just see like the tracking die. But then you can stain the gel with this chemical called Comassi blue. And so it's this stain that will bind to the proteins and absorb light, so it looks blue. And you can see where the protein is. And so that's the idea with Sds Page. So to review, we need the Sds is important for doing a few things. It helps denature the protein. So it kind of like slithers in and helps like push things apart and denature it along with the heat. It also keeps the proteins linear and soluble once it's like globbed onto the whole protein. And it gives them the negative charge. So those things are really important. There is one complication. So this is going to separate, we talked about how this is going to separate the, this is going to separate the various members of the compound, because you are now just like unfolding the proteins and coating them in this detergent that makes them just like happy on their own. And so in order, this will break up most things, but there are, so this will break up non-covalent interactions. So covalent interactions are like the strong types of interactions that are like holding together the amino acid letters in the chain, whereas non-covalent interactions are mainly responsible for giving the protein, like doing interactions between the side groups. So between like these R groups, so between each other and between the backbone to help give you this structure. And so these non-covalent, these are weaker interactions. And these are the type of interactions that are responsible for most of the quaternary structure of proteins. So most of the interactions between protein chains are happening through these non-covalent interactions, and therefore you can break them up with the heat and the SDS. However, there are some linkages that are covalent interactions. And so the type this happens is in cysteine crosslinks or disulfide bonds. So here it's showing it within a single protein, but this can also happen between proteins. And so basically cysteine is one of the amino acids, and it can oxidize and form crosslinks. And so you don't need to worry about like the oxidation and reduction, like the chemistry and stuff at this point. Basically, oxidation is loss of electrons, reduction is gain of electrons. So basically, a cysteine, you can, when it gets oxidized, it can oxidize and form these crosslinks and then reduce and break these crosslinks. And so these crosslinks are covalent bonds. So they're actually like sharing pairs of electrons, which is what like happens in covalent bonds, but they're not as strong as the bonds like holding the different letters together. So these bonds have a special weakness in that they're sensitive to the oxidation status, like the oxidation and reduction environment. So what you can do is you can actually add reducing agents like DTT or beta-mer ethanol. And what these are actually going to do is they're going to break up those, they're going to reduce those cysteines back this crosslinks back into their uncrosslinked form. And so this, you can run an SDS page, or even when we're talking about native page. So if you run an SDS page, you typically add a reducing agent. So you're gonna break up the crosslinks as well. However, you can also run it without the reducing agent. So that would be denaturing, but not reducing conditions. But typically you do reducing and denaturing. But if you say you like suspect that you might have a disulfide bridge, you can test it with and without. And so, yeah, so then once you stain your protein, your gel, you see these bands corresponding to the location that the protein was in when it stopped. And so there are different versions of stains. These are both versions of like chromacy blue staining. So this is like the classical and like instant colloidal forms. Okay, so that's the SDS page, but let's talk about the native page. So with the native page, our proteins are staying together. Our complexes are staying together. You can make page gels with like various amounts of like meshiness shown by twiddling with the amount of like the cross linkers and stuff. You can get different percentage gels. So higher percentage gel is going to have like a tighter mesh, and it's better for separating really small things, which could just like all easily travel through the big holes. And so you're not really going to get good resolution or separation of those. But for bigger things, you want bigger holes. But because with the complex and all this stuff, you can have this wide range of sizes. And because now you also have this like shape to contend with, and not just because remember, the SDS is going to like unfold and linearize them all. But here with the native page, you're not linearizing them. So they also have a shape to contend with. So now instead of like slithering through basketball hoops, you're kind of just like dropping through hoops somewhat. And so you want to use a gradient gel. So sometimes often like a 4 to 12% or something. And here, the pore size is going to gradually get smaller as you go down. And so the complexes are going to move until it gets too tight for them to move easily forward. And so if we can do this in various ways. So if we can start with the proteins, like purified proteins, and mix them together and see if we see a change in the position, like a gel shift. Or we can take like a sample of proteins from like a lysate or something. So like a cell, you break cells open and you want to see what proteins are hanging out with each other in a sample. And so you can then run them, and then you have a couple of options. So one of the options is, so if you just run it and stain it and look at it and take a picture and be like, cool, I see a shift, or I don't see a shift if you're starting with purified things. Or you can just be like, oh, this looks cool. But typically you want to do something with it. So one of the things you can do is you can do a Western blot. With the Western blot, what you're going to do is after you do the electrophoresis, you're going to transfer the proteins. You're going to do like another electrophoresis, but so you went down and now you're going to go out, and you're going to go out onto a membrane. And so you're transferring the proteins onto this membrane, and then you can probe them with antibodies. So these little proteins that have specifically bind to a protein that you're interested in, and then you can see where that protein is in the gel. And so this is helpful if you know you need to know what you're fishing for. And with this page method or whatever, you might want to be probing, like you can probe for one thing and then wash that off and probe for another thing, and then see if they or your signal was at the same place, or you can look at the size of the shift or whatever. Another thing you could do is you could actually, there's different methods. So you could actually either just like cut out the band and send it for mass spectrometry, which will identify by the peptide sequences, like what's in the gel. Sorry for like switching back and forth between slides. I'm just trying to find the one I'm looking for. And then there are methods where you can like diffuse it out or electrolute it out, or I don't know, there's various different things you can do to like isolate the proteins after this point. But another thing that you could do is take these and separate them further. And so native page is often used as the first dimension of like a multi-dimension page. And so there's one form of 2D electrophoresis where you do native page followed by Sds page. And so 2D electrophoresis, there's different forms of 2D electrophoresis. And so the one you might typically hear about the most is like where you start with isoelectric focusing, where you run a like this strip where it has like a charge gradient, and so you're separating the proteins based on their charge, and then you are going to you run like Sds page. That's not what we're talking about here, but you can do isoelectric focusing like after native page or that sort of thing, and so they can do that. Or another common one is to do Sds page afterwards. So basically the idea here is that you have this protein, you have these complexes, and now if you were to like cut out the slice and now you're going to denature it. So now you add the Sds, and your reducing agent, and then you will, then you can turn that, embed it horizontally across the gel. So you actually can like pour the gel like around it so it polymerizes around it, and you like leave one lane for a ladder, and then you run the sample, and the proteins that were in the same complex during the first part are going to run in the same column, whereas the protein that was not in the complex, so the proteins that were together in the complex are going to run together, and proteins that weren't complex are going to run separately. And so now these are going to be in different like columns, but you don't have like distinct columns, like because you don't have wells like you had normally. And so what you get is more like smears across the thing and just like dots and stuff everywhere, because there's no like specified columns or whatever, and no bands because you don't have like this wells. And then you can do things like you can do the blotting or mass spec or all of that stuff that you could have done before, but now you have better separation. And so you can get cleaner results. But what if your protein is not, doesn't have a negative charge? So we've been talking about so far is still going to apply to this, to when you're doing a blue native page, most of the stuff, but you're just going to add this dye. So basically with the Sds page, remember the Sds was doing several things for us. It was doing denaturing it, keeping it soluble, and importantly, making it negative. And if we don't add Sds, we're not just keeping the complexes together, we're also not doing anything about the protein's charge. And this can be important because proteins have different charges. And so the protein's charge is going to depend on its like combination of amino acid letters. And so there are some amino acid letters are usually negatively charged, and some are positively charged, and different proteins have different combinations of these letters, and so they'll have different charges. And I did a lot more on this in the other day's posts. You can check out. But basically, this is also going to depend on the pH, because at a lower pH where you have acidic conditions, this means that there are more protons available. And so the protein can take up this positive charge. The amino acids that can be protonated are more likely to be protonated. So the pH is going to depend on or the charge of the protein is going to depend on the pH. And there's a specific value for each protein called its isoelectric point, or PI, which is the point at which it is electrically neutral. And so overall, like net neutral. So much more on the other post, but just know that if you have a high pKa, we tend to call these proteins basic, and they're typically positively charged. Whereas if you have a low PI, then they are typically negatively charged, and we call these acidic. And so if we have like an acidic protein, that's not a problem for page, even if you, for native page, because you still have a negative charge. And remember, the negative charge is what's important for getting the proteins to move through the gel, because you have this this charge gradient and the opposite charge to attract, and so the negatively charged protein is going to be propelled through the gel. But if you have a positively charged, if you have a basic protein, then your protein isn't going to be attracted to the bottom, it's going to be attracted to the top. And so you're either going to get like stuck in the wells, or it's actually going to come up out of the wells, and you don't want either of those things to happen. So are you out of luck? Not necessarily. And so one way that you can do this is with this thing called Blue Native Page. And so here you're still keeping the complexes together. You're not adding SDS, but you are adding Comassi blue dye. And so the G250 form specifically, but this is going to bind to proteins, just like it binds to proteins to stain them, or let you visualize them when you're staining a gel. But it's going to, it binds more gently than the SDS. It's not like denaturing, it's not slithering in them or anything. It's just kind of lightly binding to their outside. And it's going to give them this negative charge that they need to make the protein negative. But because these reactions are weak, you have to actually put this dye into like the buffer so that it is constantly replenished along the protein's journey, and giving it that negative charge it needs to travel.
Just looks so cool. It's like, so Blue Page looks kind of freaking awesome. So the reason why it's kind of like you have that band is because you start it with the dye, and then you replace it with like fresh undyed buffer, so you don't like over dye it and stuff. And so I'm running it, and my protein's somewhere in there. Hopefully I will be able to see it. And I think that I will post separately. I have a video